Good afternoon. Welcome back. Today we're going to talk about viral ecology. This is a brand new lecture in this series, never before given. And it will certainly be here for a long time to stay in this course. I think it's very important. It's something we haven't touched on before. The whole idea for this lecture, I ascribe to this meeting I've been attending. I've actually attended twice. It's the Giant Virus Biology Symposium, which takes place in Tegernsee, which is a couple of hours outside of Munich. And, you know, this is just a fabulous setting. It's this little castle called Ringberg Castle, and small meeting, as you can see, less than 100 people. And some of these people are virologists. Like that's a virologist, Jim Van Etten, Nelselde Rich Condit, and a few others. But many of these people are ecologists. Many of them are, in fact, marine biologists, because it turns out that these giant viruses are plentiful in the sea. And when you study giant viruses, you get into questions of ecology, and some of which we'll touch on today. So I got inspired because these individuals are not interested in viruses because they make you sick. They are interested in viruses because of their impact on the environment, on the ecology of the planet. And, you know, I went to this meeting a couple of years ago. I went last November, and I said, this is so important. It's grown as a field. We have to have it in this lecture series. Let's see, there's one person here who has helped me a lot with the slide, Steve Wilhelm. He's back here. He's in Tennessee. He studies freshwater viral ecology. And he was a student with Curtis Suttle here, who is in Vancouver, who has done a lot of work on ocean viruses. A lot of interesting people here. I'm going to show you the program because there's some just different stuff that you would normally see. Here, for example, Alexandra Warden, a sea of giant viruses. Diversity of giant viruses infecting haptophytes. Here's a cool one. This guy, Greek Stewart, works at the University of Hawaii. It's nice. Here's another virus we'll talk about today, Steve Wilhelm, AAV particle. Jim Van Etten has studied chloroviruses his entire career. We're going to talk a little bit about that and a few other things I wanted to show you. See here, distribution of giant viruses suggests a complex ecology, individuality, and host-virus interactions. So the point here is that you get inspired to think about different things with respect to viruses. You know, I've been human disease-centric, and so this inspired me. And at the end of this meeting, I said, I have to do a virus ecology lecture. So this is what came out of that. It is certainly a work in progress, and it will certainly be modified year to year after year because this is a relatively young field, viral ecology, and it's growing. While I was there, we did a podcast, and Rich and Nels, Alexandra Warden, Asaf Vardi, and Steve Wilhelm, and we will talk about each of their viruses today. You can tell it's Germany, right? They have beer here during the talks. So ecology, of course, is a study of the relationships of organisms, not only to each other, but to their environment. You know, so we often talk about viruses in cells and culture with no regard to the environment. And this is really different. And so that's why I wanted to put this in the course. And the way I view it, I view it in two ways. I view there's some microecology here. I don't know if this is the right term because I'm not an ecologist, but this is how I'm thinking about it. I'm thinking about the interaction of viruses within their hosts. So a virus comes into your respiratory tract. It moves up and down the tract. It may go below the basement membrane. That's ecology, but it's microecology, right? Because we're looking within a host within cells. And there are definitely influences of the environment within you. And if the virus is in the GI tract, it's going to be a different interaction. And we have talked much about this in this course, especially in the second half of the course. That's been a major focus. I'm not going to talk very much about that today. I want to today focus on what I call macroecology, and that is the bigger picture. And there are two separate divisions of that I view. One is looking at ecological disruptions that lead to zoonotic spillover, because every spillover. Every human virus came from an animal virus, and that was usually because of a perturbation of the environment in some way. So I think those are ecological issues for sure. And then there are what most of those people at the giant virus meeting work on, the global effects of viruses. What are viruses actually doing out there when they're infecting plants and unicellular organisms and so forth. So I think these are both ecology, all three things, the micro and macro, the two subsets of macroecology. They're all ecological issues. So I want to give you an overview. So this is going to be a survey today. I'm not going to go into a lot of mechanism. In fact, there isn't a lot of mechanism in terms of molecular biology, but that's fine because it's fascinating nonetheless. I want to talk first about ecology and emerging viruses. And here, of course, we're talking about disruption or invasion of ecological niches that lead to cross-species transmission. As I just said, every human virus, by which I mean viruses that transmit among humans regularly, as well as zoonotic viruses where every outbreak is a spillover. So human viruses and zoonotic viruses, each one, their origins can be traced to invasions of ecosystems and disruptions of ecosystems. For example, and we've talked about this, I just want to put this in a framework, a formal framework. Measles virus was a cow virus, rinderpest, that we acquired when we started putting cows together. We disrupted the cow ecosystem. Cows were living in the wild. We domesticated them. We put them behind fences and grew them in large numbers, which is totally not the way they originated, right? And we got their viruses, but it all comes from us making a disruption. That's how I view it. Ebola viruses, we go in the forest and do bushmeat hunting, and that's not a modern thing. Of course, humans have been hunting bushmeat forever, but it's still an ecological disruption. Nipah virus, we're going to talk about in more detail today, a disruption of the bat life, and SARS coronaviruses. Some disruptions that lead to interactions of humans with bats. And in the first SARS, of course, it was the meat market, the bringing in of animals from the surrounding countryside and putting them in a meat market, and that causes the crossover. In the current SARS-CoV-2, we're not quite sure how it happened yet. But before we talk about spillovers in terms of ecology, a little bit on some general principles, microecology, if you will. I showed this slide to you, I think the first lecture of this course back in January, to tell you that we have a virome. So virome means all the genomes of all the viruses in us. And, it's just one of these ohm words, genome, proteome, kinome. I heard someone say that today. The kinome is all the proteins that are phosphorylated in cells, metabolome, et cetera. You get the picture. So virome is all the genome. So what you do is you'll take a swab of skin and extract the nucleic acid and sequence it and look at all the viruses. Respiratory tract, urogenital tract, digestive tract, same idea. You're just doing metagenomic sequencing, which means you're sequencing all the nucleic acids and you're focusing on the virome. And this is a new technology, of course, 10 years so ago developed. Fabulous technology, lets us have a window on things that we never had before. And in fact, a lot of the work today comes from metagenomic analysis. But through it, we know that humans have a DNA and an RNA virome. And these are the number of different kinds of viral genomes we see in these different areas. And there are lots of them. And we don't know if these are infectious viruses. Some of them might be, some of them might not, because again, we're just sequencing. But of course, the implication is that we have a lot of viruses in us. They're obviously doing something. They're not all making us sick. And the question is, what are they doing? And what is their interaction with us? So this is like the microecology question. We don't know. We don't know the answer to this. We have no experiments that would tell us anything about the function of the human virome. I'm assuming in the future we'll be able to do some kinds of experiments. The other day we were talking about using CRISPR to remove herpes viruses from latently infected ganglia. Probably people are going to want to do that at some point. It should be doable. And then we'll see if when you take away the herpes simplex virus, if it's a problem or not. I don't know if it is a problem. So there are viromes in other organisms, of course. And in plants, it's been called the phytobiome. And so this is all the viruses in plants, but not just plants, but everything. And that's why it's the phytobiome and not just the plant virome, because that is a thing, of course, the viruses that are existing in plants. And there are tons of them. We're going to not talk much about them here today. It's a shortcoming, but in future years, I hope to do it more. So the genome, not just viruses, but everything that's interacting, microbes in the soil, fungi, nematodes, insects, other plants, animals. So that's the phytobiome. And I'll show you some examples of at least how the viruses interact with plants today. So every organism has this. And, that rabbit has its own virome. And it's doing something, but it's barely been studied. And every animal on Earth has it, and hardly any of them have been studied. Some bats probably more than any others because they're sources of zoonotic viruses, but everything else has been undersampled. It needs to be sampled. It's really hard to get funding to do field work like that, and a lot of people don't want to do field work. I've talked to a number of people on TWIV who lament the lack of field work. But, it's hard, not just getting funding, but you have to go somewhere for months at a time. And if you have a family, that's hard to do and so forth. So it's getting to be less and less done. But now we need it more and more, especially with the ability to look at omes. We know that there are all kinds of virus-host symbioses that are outgrowths of having viromes. And some of them are summarized here. For example, some of them are antagonistic, the kinds that we've been talking about in this course, dealing with humans where the virus causes disease. We call that antagonistic. And here we're looking at a virus, tomato bushy stunt virus. It affects eggplants. It's obviously causing them to have problems. It's making them sick. And this is, if you're growing eggplants to sell, this is a problem, obviously. So you have to figure out ways to control this. So that's an antagonistic relationship. Symbiosis is neutral. It just means there's an interaction, right? Then it could be commensal, viruses with no apparent impact. The human virome, a lot of those viruses, we say are commensal, but maybe someday we'll find out that some of them are mutualistic. They help us. We help them, of course, because we give them the host. But right now we call them commensals. They're just living there. Then there's some conditionally mutualistic viruses. This is quite interesting. Under certain conditions, the virus is neutral. Under certain conditions, it may be beneficial. But the host is the rosy apple aphid. And when infected, this particular virus, which I'll bet is called rosy apple aphid virus, it induces formation of the winged morph. So normally, the aphids don't have wings. They're feeding on sap. They get infected and they develop wings. And these are smaller. They're less fecund, so they give rise to fewer offspring. But they can move to a new plant if the plant gets overcrowded. Too many other aphids, they can move because they can fly, and these other ones, they can't go very far because they can just walk. So helps that one with the wings to get somewhere else. So it's conditionally mutualistic. So in other words, if there's no crowding, this aphid's not going to go anywhere, so it doesn't help them. And then we have mutualistic, straight mutualistic viruses here. A bacteriophage infects Vibrio cholera, a bacterium, and the phage brings in a gene for a toxin, a cholera toxin, and that allows the bacteria to invade the host gut. Otherwise, the Vibrio is not a dangerous organism. It lives in the environment and it's fine. But when it gets this phage, then it can invade your gut. So this gives it some way of moving into a new niche. And then finally, the kind of symbiosis we've talked about, which is this author of this particular study called symbiogenic. And this is when you take something from a virus, in this case, the exaptation of a retroviral gene to form the placenta, which we talked about. The envelope gene became an endogenous, part of the endogenous retrovirus in marsupials probably many millions of years ago. It was taken by the host to be able to form the fused layer of cells that gives rise to the placenta. Very cool. So there are lots of these symbioses, and really we've just scratched the surface. We really can study a lot more. And what's missing, of course, is a lot of mechanism, how many of these work. And we talked about beneficial viruses as well. So those would be the mutualistic ones. And we talked about how a virus within a fungus allows a plant, a grass, Dicanthelium, to grow at high temperatures. So these are grasses that would grow around the hot springs, say in Yellowstone National Park. They're able to grow because they have a fungus in them, Curvularia, but the fungus also has a virus. Both are required, so it's a three-way mutualism. If you take out both or one, you can take out the virus from the fungus, and the fungus alone, the plant will not be able to grow at high temperatures. So this is a wonderful example of a mutualism. And this is very widespread in the plant world. And the mechanisms are really not known. And that's why I say this is a survey lecture, because I can't tell you what the mechanism of the virus and the fungus making this plant able to be thermotolerant, basically. And here's an example of a virus that confers drought tolerance. Many plant viruses make the plants resistant to drought. Right, so here, if we don't have a virus under conditions of stress like drought, the plant dies. But when the virus is present in a symbiosis, a mutualistic symbiosis, then under conditions of drought, the virus-infected plant will survive. And this is being explored because, wild plants have lots of viruses, and they're quite tolerant to all sorts of adverse conditions. But the crops we grow for food, those are monocultures. They're derived by breeding over the years, and they're all derived from one source, and they don't have viruses in them. And some people believe that we should put certain viruses in them to make them more robust. That's an area of interesting research. So first I want to talk about two stories of virus emergence with an ecological bent, right? I want to take a little different turn from last time, where we mainly talked about the viruses that are causing certain zoonotic infections. And here why? What are the factors leading to emergence? So here's Nipah virus, which you remember caused an outbreak, the first outbreak in Malaysia in 1998. And this was an outbreak, of course, in pigs in the Perak region of Malaysia, which eventually spread south to another part of Malaysia and then eventually to Singapore, all by transport of infected pigs. And the millions of pigs got infected, and many developed respiratory and neurological disease. And some of those pigs passed the virus on to humans, 105 of whom died. So, and then this virus has subsequently caused outbreaks in other countries as well. The conditions that led to the outbreak here were corrected. But here's what we think happened. And I'm giving you this as an example because it's a really nice analysis of what we think happened. First of all, in the 70s through the 90s, the years before the outbreak, a lot of deforestation going on throughout Malaysia, not only to get wood, but to clear the area for industrial strength plantations, to grow crops for people, but on many, many, many acres. Take out all the trees, you have to do that. So there's deforestation. This reduces the habitat of the fruit bat, which of course is the reservoir for Nipah virus, Pteropus vampirus. So we're destroying the habitat. And then on top of it, a lot of this deforestation is done by slash and burn. So they cut down the trees and then they set them on fire. So they burn and then they plow in the ash into the soil. But all this burning makes atmospheric haze. And in fact, in the months before the outbreak, it was noted that this haze blanketed much of Southeast Asia just before the outbreak. And so that probably had a negative effect on the bats as well, maybe driving them away from their normal habitat. And finally, the third condition, there was a drought in this time, just before the outbreak, driven by the 97-98 El Nino, this southern oscillation that can drive drought, and so there wasn't much rain. So the outcome of all this is a reduction in the availability of flowering and fruiting forest trees where the flying foxes live and forage for food. So we're taking away the trees, we're making it hazy so they can't see where to go, and they don't really want to fly through the smoke, I suppose, so they're diverted, and they look elsewhere. And so where did they look? Well, these piggeries that had been built, and these are big farms with many, many pigs on, hundreds of thousands of pigs. They were built next to fruit orchards, and so the flying foxes go into the fruit orchards because we're chasing them out of their natural habitat. And then the piggeries are open. That's another problem. You have to consider now biosecurity when you build animal farms, because otherwise you have encroachment of bats and rodents that can bring in viruses, and it wasn't considered. These were just open piggeries, and so the bats could just fly in and contaminate the pigs. And so that's the ecological disruption that led to this outbreak. And, in every outbreak or spillover, there's some kind of disruption, as I said. It just, in some cases, we just haven't figured it out, but there always is something, anthropogenic activity, that leads to the outbreak. So in the end, it's our fault, really. So here's a summary of this web of Nipah virus emergence. There's your fruit flying fox or fruit bat. Adorable, right? These are very big animals, by the way. These are the natural reservoir hosts of the virus. So we have deforestation driven by needing land that reduces their habitat. It reduces their food supply. We have a drought, which further makes it shortage of their food supply. Drought, trees are not going to have as much fruit. We have forest fires that we're making to get rid of the forest, and that creates the haze, which further impacts the food supply. And so the bats migrate. They migrate away from where they would usually live. They're forced out by these conditions. And then they happen to go into the fruit orchards, which are built next to the piggeries, or the piggeries are put next to the fruit orchards. And the pig farms are open, pigs get infected, and humans get infected. So that's the story with Nipah virus emergence. Here's a summary of that in a broader sense to take into account the subsequent outbreaks. And it is all about ecology, obviously, because we have bats and pigs and humans living outside, and their relationships determine how the infection goes. So we have the bats that are foraging in fruit trees on pig farms, and again, they're foraging. They normally wouldn't do that. They would be in the forest, but we took away their forest, smoked it out, burned it out, etc. El Nino. So the bats are here. The pigs are exposed to fecal material dropped by the bats. The pigs get infected. It spreads, and then people get infected. So that's the one kind of outbreak in 1998 that can be controlled because you can cover the pig farms, use biosecurity so that the bats don't contaminate the pigs. And this is no longer happening. However, we do have infections ongoing with Nipah virus in India and Bangladesh, and they're linked, as I said last time, to the collection of date palm sap by people who like to drink it. They not only like to drink it, they like to ferment it. It's called toddy, I think, and it's an alcoholic beverage, but it all starts with being collected, and the bats come at night, and they feed, and they urinate or defecate, and they put virus in the sap, and people drink it and they get sick. And then, you care for the sick people, you prepare their body for burial, which is traditional, and then the family members get infected. Now, this was solved by a very low-tech solution, which was just to put wooden lids on these sap collection containers. If you are ever in Washington, well, nobody's going anywhere now, right? But when we travel again, the Smithsonian Natural History Museum, the one with the dinosaurs, they have an exhibit called Outbreak. And it's great. And they have a little scenario of Nipah with a palm tree and a date palm sap thing and a bat hanging from the ceiling. It's really good. We did a TWIV there a couple of years ago. It's awesome. Really good. And then, as I said yesterday, there's some infections that occur that are not linked to date palm sap. Because you're going to ask people, have you drunk date palm sap? No, but you still got sick. And so there must be some spillover from a bat And horses are implicated perhaps as intermediates in some of these outbreaks that have been studied. But again, we're disrupting the ecological balance. We're living next to where the bats are. Maybe we're driving them out of other areas. So this needs a Nipah vaccine, I think, and that is in development, as I said. All right, let's look at one more. There's so many examples of these are fascinating, but I don't know, you get the idea of what I'm trying to tell you. But just this one is, it's also involving agriculture. Really, it's a big driver of emergence. And this is a disease called Argentine hemorrhagic fever, called by Junin virus. And so after between World War I and II in the Argentine Pampa, these very fertile regions of Argentina, they wanted to convert it to farmland to grow maize. So there are a lot of weeds there, right? So they treat the area with herbicides. It kills the weeds, and then you can grow corn or maize. But as the corn is growing, there's this new grass growing in the shadow. It's a shade-tolerant grass that wasn't there before, growing in the shadow because you've gotten rid of all the competitor weeds. Now this one's moved in. It can grow in the shade of the maize. Nothing else can grow there. And this grass is favored by this mouse, Calomus musculinus. It's never been there before. It moves in, becomes the new dominant mouse. And this mouse happens to be a reservoir of Junin virus, which is one of these arenaviruses that have ambisense genomes, enveloped RNA viruses with ambisense genomes. So this mouse is the natural reservoir. It doesn't get sick from it. It sheds it. The mouse population goes way up, and then, of course, it goes into homes, and people get Argentine hemorrhagic fever. It was first described in 1953, and then 5 years later, the virus was isolated. They called it Junin virus because these outbreaks happened outside that city in Argentina, Junin. So another example, we mess things up because we're just thinking about ourselves. Well, I guess we need food, fine. But now you can grow food indoors. You don't have to plow lands and cut down trees. You can do indoor farming, and it doesn't need dirt. It doesn't need much fertilizer. It just needs water, hydroponic. It's great, and I hope we convert to that so we don't have to do this. I mean, one of the things we'll talk about in a bit is algal blooms in the oceans. They often happen when fertilizer runs off from farms. Farmers put down fertilizer. It rains, it runs off into the ocean. You get blooms of algae. Some of them are toxic. And we don't need to do that with indoor farming. But that's another course, not virology. All right, this is another slide I showed you in the beginning that just emphasizes how many virus particles there are on Earth. Over 10 to the 30th just in water. And the weight exceeds elephants by 1,000-fold and put head to tail 100 million light years into space. Staggering numbers of viruses. Now, I want to talk about what these viruses are doing. What are they, and what are they doing in the world's waters? This is another slide we showed. I'm sure you don't remember this. A long time ago, Lecture 1. There are more viruses in a liter of coastal seawater than there are people on Earth. Over 7 billion, well, way more than 7 billion. And if you look at biomass in the oceans, most of it is prokaryotes. But if you look at abundance by particle number, most of it is viruses, 94% viruses by particle counts. And this is only something we discovered, 15 years ago by Curtis Suttle and his associates, that person I pointed out to you in the German giant virus meeting. Here's how they were discovered. People started doing electron micrographs of seawater, but that's not very efficient. And then Curtis had the very clever idea to just stain water with cyber green. This is a dye that binds to nucleic acid, and it fluoresces green under UV light. So he took seawater, he filtered it through a 0.2 micron filter to get the gunk out. And of course, you're going to lose a lot of giant viruses that way, but you can look at those with other filters. And then you stain it. And so you get all these dots, right? What are they? Well, the big ones are bacteria. But if you do a preparation of a bacteriophage, T7, you do the same staining with cyber green, you see these little dots. And that's what these little guys are, all in the back. So you can look in waters, any water, seawater, freshwater, coastal, pelagic, whatever you want. You can look and see how many, you can count the particles. So you can know how much or in the field, what volume is in the field, and you can count the particles. And so in seawater, we have about 10 million virus particles per milliliter. And that's in part how we get that 10 to the 30th number, because we have a rough idea of how much water there is on the planet. And then people then started doing this all over the place, right? Not just Curtis Suttle is from Vancouver. He could just go out his door and get water. But then people took ship expeditions all over and got water and did that. And here are sample estimates for virus-like particles and bacteria in freshwater systems. Lots of different studies you can see here in different places: Germany, Canada, Antarctic lakes, France, Sweden, Senegal, Italy, etc. So they sample water, different environments here, and they put the cyber green in it. That's called epifluorescence microscopy. And so they, and you see they have some brackish, they have some saline, some fresh. Virus-like particles. So they're calling them virus-like particles because they don't know if they're actually infectious. Times 10 to the 10th per liter. So you get varying ranges, right? You can see some very abundant and some not so much. And then you can look at the bacteria as well, because it turns out the bacteria are hosts for many of these viruses. Every water on the earth basically has viruses in it. Some of these lakes at the poles, the poles have lakes, and especially in the spring they thaw, and they have very low complexity microbial communities, so they don't sport a lot of viruses in them. So you can see this is pretty low, but compared to Mono Lake in California, which is a weirdly acidic lake in California, but it still has microbial communities. So again, what do these viruses do? I'm going to tell you a couple of stories of what they do. So first of all, most of them are thought to infect heterotrophic bacteria, and they also infect cyanobacteria. These are the bacteria that make a lot of the oxygen that we have in the planet. That oxygen is made by plants and cyanobacteria in the ocean. So there are lots of viruses of them. And then many of these viruses infect eukaryotic phytoplankton. And those are among the things that cause these large ocean blooms, like this one here. And the virus can actually collapse the blooms. So here's a bloom. I think this is the boot of Italy. This is huge. These thousands and thousands of square kilometers. You can see this is a photo from space, actually. It's a satellite photo caused by a particular phytoplankton that we'll talk about in a moment. But these things rise and fall. You could see them coming up and going down, and there are reasons why they come up and come down. But viruses can impact these. So that bloom I showed you is caused by a single-cell coccolithophore. The coccolithophore is a photosynthetic plankton. It's found everywhere in all the oceans. Single cell. And that's a picture of it right there. Bar is 2 microns. It's one of many parts of the marine food web. All kinds of plankton and other things are part of the food web, of course, but this is part of it. So things feed on it. It's pretty down on the food chain, right? It's probably a primary producer because it's photosynthetic. It makes blooms, and it makes them in nutrient-depleted waters after the reformation of the summer thermocline. The top layer of the ocean gets warm, and the bottom is still cold, and that's the thermocline, and that reforms in the summer, and these blooms tend to form then. These are really amazing organisms. This looks like someone made it, right? But it's a plankton, and it's covered with calcite discs. That's what these are. Gorgeous calcite discs. And these are made up largely of calcium carbonate. And when these things are huge numbers, right, in the oceans, those blooms form, and they can constitute 75% of the microbial community at that time when the bloom is there. And when they die, they fall to the bottom of the ocean, and they make piles and piles of calcium carbonate, which have come up. And these white cliffs of Dover are mainly made of dead Emiliania huxleyi. It's all calcium carbonate from these calcite discs. Really amazing. And the bottom of the oceans are full of them. Any time you go inland and you see calcium carbonate that's been pushed up from the ocean floor, it's coming from this. So this is a big source of calcium carbonate, but it's also a big source of dimethyl sulfide. And this is a gas that's produced and comes out, and it's converted to other compounds, which then induce cloud formation. So when there is a big bloom of E. hux, it produces a lot of dimethyl sulfate, which comes out of the ocean and rises. It's converted and causes cloud formation. Now, these are photosynthetic plankton, so when there's sun, they multiply. And then when the clouds come out, the blooms recede, okay, because there's no sun. So it's like they're regulating their own formation. It's really remarkable. I mean, this is why ecology is great. Anyway, there is a virus, of course, of Emiliania huxleyi. It's called EH virus 86. And by the way, so this would normally be italicized, Emiliania huxleyi, but when it's a virus, you don't italicize it any longer. That's the rule. So this is a coccolithovirus because these are coccolithophores, and it's a giant virus. It is a giant icosahedral DNA virus, the big T number, and it has a linear double-stranded DNA genome, 4 to 415 kilobases. I mean, it's not the biggest, right? We've got millions of bases of viral DNA, but it's pretty big, and it encodes 472 proteins. And so it consists of this capsid, and around it is a membrane, really cool membrane, and then we have in it the DNA. And this is what we call a nucleocytoplasmic large DNA-containing virus. This is a kind of grouping of viruses that they're large and they replicate in the cytosol and in the nucleus of the cell. And some of them are just cytosolic, like poxviruses. Poxviruses are members of this, but many of them need both the nucleus and the cytosol for replication. Anyway, so this was isolated, and it's studied in a number of labs. So Saad Vardi, who was on that TWIV, studies this virus and its effect on E. hux. You know, it reprograms E. hux to do weird things. And he even developed a plaque assay. Really cool. So what do these viruses do? So that E. hux virus, by the way, this kills the bloom. So the combination of clouds and this virus kills the bloom. It goes into E. hux, and there must fit in between the gaps here. It gets into the cell and infects it, and it's a lytic virus. It kills it, and the calcium carbonate, the calcite discs fall to the bottom of the ocean, and the bloom subsides. So these viruses are always around and maintained in the waters, and then as the bloom increases, they have lots of hosts to infect, and it then suppresses the bloom. It's very interesting dynamic. People don't really study this much because these viruses are floating around in the water all the time. They have to encounter hosts, and it's a really interesting situation. I guess not much different from an aerosolized virus that has to infect animals. Anyway, what do these viruses do? Let's talk a little bit about what they do. They are catalysts for biogeochemical cycling, and we didn't know this before 15 years ago. It's just so cool. So we have here in the ocean, this is an ocean cycle. Here we have phytoplankton, like E. hux and others. There are many others. So that's the primary producers. They're at the bottom of the food web, and they're eaten by grazers that swim around and eat. There are all kinds of grazers that will eat these. And then the grazers are eaten by carnivores, like fish. So that's your different levels of your food web. And we always thought that, what happens in the ocean, the grazers die and the carnivores die at some point, and they become organic matter. So OM is organic matter, and P is particulate, and D is dissolved. So it depends on how degraded it is. It can be particles or dissolved organic matter. And this organic matter is then taken up by heterotrophic bacteria. They use the organic matter to grow, make the molecules they need to grow, and they produce carbon dioxide. So that's how we thought it worked, this biogeochemical cycling. But now we understand that viruses actually shunt this. They catalyze the movement of nutrients from organisms to these pools because they kill phytoplankton in big numbers, and they kill bacteria too. They make a lot of PDOM. So basically, viruses short-circuit the flow of carbon and nutrients from phytoplankton and bacteria to higher trophic levels. So this pathway is to higher trophic levels. Viruses are shunting that. They're short-circuiting it, if you will. and they're making more PDOM. And so you get more carbon released. And this decreases trophic transfer through the food web, but you get more carbon dioxide or carbon in the end. And again, this is something we didn't realize, and it's essential. It's most likely essential. And just as just carbon we're looking at, they also participate in sulfur and phosphorus and other biogeochemical cycles as well. And here's a more detailed version of this carbon mass on the planet, besides what's happening in the oceans. So again, viruses can affect the efficiency. The biological pump, by the way, this is a word for sequestration of carbon from the atmosphere to the ocean and to ocean sediments. That's the biological pump. So it's always happening. The ocean is taking carbon from the atmosphere and sequestering it. Of course, when it goes to the sea floor, it can be sequestered for millions or thousands of years, of course. And so here we have sources of carbon in the atmosphere. The atmosphere has about, at any given time, about 800 gigatons of carbon. It's a lot of carbon. A ton is a metric ton. And you add about 4 gigatons a year to that. And by anthropogenic activities like deforestation, we burn forests, and that adds carbon to the atmosphere. Fossil fuel usage adds carbon. And, we shouldn't really do these things. We don't have to. But it's cheap. People take the cheapest route, always. It turns out that viruses mobilize enough carbon. The carbon that they mobilize per year is about 20% of the atmospheric content. So 20% of this is donated by viruses. And here are some of these. This is the biological pump where carbon comes out of the ocean, then it's recycled back in, and a lot of it stays in the waters, but much of it sinks. And, that's because plankton, which constitute about 5 gigatons of carbon, they die and they can sink, or they can make this, organic matter, the particulate and dissolved organic matter. Viruses can lyse them, as we just said, giving rise to particulate organic compounds and dissolved organic compounds. And, some of those are used by plankton for growth. Some of them are irradiated by a UV and converted to other compounds. Many of them come out of the ocean and are recycled back to the, so out go 90 gigatons a year of carbon, and in go 93 gigatons a year. And so the viruses participate in helping to release this carbon much faster than it would have been done by that primary food web that we talked about there. So, and then a good amount of this, 3 gigatons a year, sinks to the bottom of the ocean, and we don't see it for ages. Right, so that's what viruses do. They affect the efficiency of this pump. In the ocean, there are 10 to the 29th virus infections a day. And that destroys 20 to 40% of marine bacteria and releases 10 to the 8th to 10 to the 9th tons of carbon. Really, really remarkable. So that's what viruses do. And how do we know a lot of this? Well, there's an amazing... People go on trips and sample the ocean. And one of the coolest ones is called the Tara Oceans Expedition. And this was conceived and run in 2009, 2013. It's one boat here, the Tara, and there's the size of the thing. And this went out for this whole period on a number of trips, but they did one long one, which was well over a year. And they did this around the globe, this pathway, all over the place, sampling all the oceans. And they want to see what's there. And they have all kinds of tech on this boat. They have, microscopes for looking in the water as you're sampling. They have flow machines, flow cytometers on board. And what they do is they collect things in nets like these, with different mesh sizes up to this, from the surface to 1,000 meters down. They have pumps that filter water through different sized filters, depending on what you want to get. Here's an Emiliania there. And then they have these these Niskin bottles. Each of these is a bottle. It's lowered on a cable and they can sample at different depths. They're programmed by a wire to open up at different depths and sample the waters down to 2,000 meters. So you can do all this sampling, and then you bring it on board, and you can see what's there. You can see the zooplankton, the bacteria, and these are some of the counts that they get. And you can also look at the viruses, of course, and you can take this material home and sequence it and do metagenomics to study all these organisms, but I'm talking about viruses here, of course. And this ship would go around sampling in these ways. Every 6 to 8 weeks, it would freeze the samples and ship them off to labs where they'd get analyzed. And, these are all the different institutions involved, and people take turns on this boat. But this is amazing. And these samples are available, and the metagenomics are available. You can find websites where all of this is. Anyway, so that's how we have learned what's in the oceans. All right, so from this, we can tell where the carbon is in the sea. The total carbon is 10 to the 6 tons, 1 million tons. Ton is a metric ton. It's not a regular ton. It is 4 to 12 in whales. Look at this. 2,000 to 27,000 in bacteria, depending on where you are. And 27 to 270 tons of viruses. It's just huge. This is a map of all the sequences based on where they are in the world. So it's called a heat map because you can show relative abundance of each sequence by the color, right? So the dark green is very abundant and the light pink is less abundant. So population abundance in a sample. And these are different geographic regions, these boxes. And so here different geographic regions, and these are different populations of viruses that are presumably related, right? So the abundance of one is a sample. It's based on recruitment of sequence reads to the population, and it has to be over 75%. And these are geographic locations. So you have incredibly detailed maps of samples from all over the world taken by that Tara Oceans explorer, basically. That's what I'm trying to tell you. I've told you about how viruses shape activity in the oceans just by carbon activity. They also shape potential. They do a lot of horizontal gene transfer. We don't know the true extent, but we think viruses move genes between both related and unrelated hosts. And if you look at this tree of life, the bacteria are gray, the archaea are green down here. Here's the common ancestor to both. And then you have all these red lines connecting them because these genes have been transferred from one lineage to another. They don't fit within the lineages, and we think these have been transferred by viruses. And we'd like to know things like, is that transfer important? Can we see one in progress? And can viruses actually use these? So I'm going to give you some examples of how viruses can use these genes. So here's the story about a giant virus that infects green algae that encodes key fermentation genes. And this is a story out of Hawaii. And this is from Greg Stewart's lab. He was at the giant virus meeting. So we have these lovely oceans off the coast of Hawaii. They have a lot of algae in them, green algae. And this particular one is a species called Tetraselmis. And it contains a virus, Tetraselmis virus 1, which is a big double-stranded DNA virus of 668,000 base pairs of double-stranded DNA. So this is an electron micrograph of a Tetraselmis algae. And you can see it's full of virus particles. And these are icosahedral particles, as you can see in this close-up. And so the genome of this virus, Tetraselmis virus 1, was sequenced. And among all the genes, interestingly, for the first time, they found fermentation genes. Hosts have fermentation genes, of course, but viruses were not found to before. So this is the first time. And so here are two of these fermentation genes, pyruvate formate lyase and pyruvate formate lyase activating enzymes. So they're involved in fermentation. And here are the viral genes here, and you can see that these are other hosts. Tetraselmis is the host. That's the virus TETV, but Tetraselmis is the host. So this is closely related to the host gene, and who had it first, we don't know. Right? The cell may have had it, the virus may have taken it, may have moved it around, but it's also in a lot of other related algae as well. As you can see, both of these enzymes on this phylogenetic tree. So this is an example of probably of horizontal gene transfer. Why would you have a fermentation gene? The virus will put these genes in the host and express them, and maybe it manipulates the fermentation capabilities of the host. And maybe if the host is in low oxygen environments, then the virus can have it switch to fermentation to get energy so it can continue to reproduce. We don't know the answers, but they're all very tantalizing possibilities. If you want to go to Hawaii, this is the thing to do. Here's another story which is amazing. I heard this, both stories I heard at the giant virus meeting. This is a virus of a choanoflagellate. I don't know if you know what a choanoflagellate is. It's the closest, it's a single cell, and it's thought to be the closest relative of multicellular eukaryotes. So maybe it's an ancestor of all of us. And this is a phylogenetic tree of eukaryotes. You got all kinds here. You got Emiliania, and you got flowers and amoeba and mammals, fungi, etc. In red are giant viruses that have been identified in various eukaryotes. You can see they're all over the place. And the more we look, the more we find. And the story I want to tell you is a giant virus of a choanoflagellate, the first one. That's the red star there. And this was identified, this was an ocean-going voyage. So besides the Tara Ocean, if you want to study the ocean, you can have your own boat. You can lease it, and many universities do, and they lease it to many groups. They all go out on trips, and you can collect samples. And the group of Alexandra Warden, who was on TWIV, and she did this work, they went out and they collected organisms from the ocean. They were interested in the single-cellular choanos, for example. And they brought them on the boat, and they did single-cell sorting, and then they sequenced each single cell, and they wanted to do the genome sequence, but they found in one choanoflagellate a complete viral genome, and it was the giant virus. So they got interested in giant viruses like that, and now they're studying this. So that's ChoanoVirus, and the cool thing is it encodes a complete rhodopsin photosystem. The whole system, not just the sensors, but all the enzymes to make the cofactors that you need for these to work. And these, of course, take light and they convert it to energy. Really cool. And so here, they did the genome sequence, of course, and here is a phylogenetic tree of this ChoanoVirus. They found 2 of them. It clusters with the Mimi viruses, which we've talked about before. We're going to talk about an Aureococcus virus in a few minutes. EHUX is somewhere on here. Here it is, EHV68. It's in a separate group. We're going to talk about chloroviruses too here. So 2 kind of general branches of giant viruses, the Mimi viruses and Phycodnaviridae. The cool thing is that here are all the rhodopsins. Here are the choanorhodopsins in the blue star. And you can see there are rhodopsins in some other viruses. Here's another giant virus, Phaeocystis globosa virus. And then there are marine bacterial rhodopsins, and there are archaeorhodopsins, eukaryotic rhodopsins. They're all related. So again, the virus probably stole this from a cell, and maybe it's moved it to other cells as well. The idea here is that these rhodopsins would let the virus make more energy in an infected cell in sunlit waters, though, okay? You can't be deep in the ocean because then it's not going to work. So then they took the sequence of these viral rhodopsins, VIR, and they plowed through the Tara Ocean's data. They can just get the data sets and look for hits with their viral rhodopsin, and then they could map the hits based on where the sampling was done in the ocean. They're using a DNA polymerase of a Mimi virus as a marker. Those are the black circles, and the frequency is related to the size of the circle. And then they're looking at viral rhodopsin in red. So you can see, first of all, they're all over the place, basically in every sample set that was taken on the Tara. So they're obviously widespread beyond their choanovirus. They can also look at the rhodopsins by depth in the ocean. So here is arranging the sequence sets by depth, because the ship sampled down to 2,000 meters. So here 0 to 1,000, and all the viral rhodopsins are here. The reeds are all in the top waters, which makes perfect sense, right? Because they need to get sun. And if you go too deep, you don't get any sun. And so then on the right here, you can further fragment the sequences based on the kinds of rhodopsins. The blue are the viral, and then the others are different kinds as well. So these are widely dispersed rhodopsins. They're probably doing something important to give energy, and that's really going to be interesting to sort out. But you can see how you can use these metagenomic sets to sort this out. It's really fascinating. All right, so we talked about how the E-hux makes blooms and how the virus can reduce the bloom. Here's another bloom former, and this is a little closer to home. This is an alga responsible for New York and New Jersey brown tides, Aureococcus anophagefferens. And here's an aerial picture of a brown tide, right? Makes the water brown. Just things overgrow, they cause blooms. We've only recognized these in estuaries since 1985. And there are tons and tons of algae here. I forgot to put the number down. Many millions per milliliter. There's a microscope picture of the algae there. And this, right by a dock, by somebody's house, there's a brown bloom. It's not good. It's detrimental to bivalves. It's detrimental to eelgrass. And, $50 million in losses a year. It's not huge, but it's probably going to keep spreading. And so this is studied by Steve Wilhelm, who, as I said, gave me a lot of these slides, or the material for a lot of these slides. And of course, there's a virus of Aureococcus. Way back in 1988, they were studying the first bloom, which was in Narragansett Bay in Rhode Island. Summer of 1985, they were taking electron micrographs of the algae. And look, they're full of virus particles. So from the start, they saw them, but they didn't try and culture them, but Steve's lab cultured this virus and did the genome sequence. So it's called AA virus. It's a giant virus, and he isolated it from a bloom. He took a sample of a brown tide bloom and isolated the virus. It probably regulates the blooms, and that can be shown here. Here is one of the few experiments done in the lab with these viruses where you can grow these AA cells, algae, in culture, and you can infect them with a virus, and then you can measure, in this case, they're measuring the cell count and the virus count. The virus count is the dotted black line. The virus count goes up when you infect these cells, and the infected cell count stays low. It's killing the cells, basically. In uninfected cells, the cells multiply. So this has the potential to regulate the bloom. Here it is, Aureococcus virus, near the ChoanoVirus, right? Very close. And again, analysis of the genome, which he has sequenced, shows this has acquired genes from a variety of cellular sources. So these are moving genes around as well. And these are just a few examples of that. There are many more examples of these viruses moving genes. The more you sequence, the more you find examples of them. And so the last story I want to tell you is a very interesting virus called Paramecium bursaria chlorella-virus. This is actually the first giant virus. Jim Van Etten over at the University of Nebraska was a pioneer. He studied this for ages and was the first to study these large DNA viruses, followed by Mimi viruses and many others. But this is a large icosahedral double-stranded DNA virus. The genome is 331,000 bases in length. You can see the icosahedral particle there, a nice colorized EM of the particle. In the inside, there's a distinct particle containing DNA. Here it is stuck on a cell, probably starting to enter, and here are some particles in a cell. Now, this virus infects chlorella, which is a kind of algae. The chlorella is a symbiont of the protozoan Paramecium. You probably remember Paramecium bursaria. You probably worked with those in grade school. There's a Paramecium, right? And you see this one's full of chlorella algae. So this virus replicates not in the Paramecium, but in the algae within the Paramecium. And that, of course, and you can see these Paramecium have cilia, right? Fuzzy. So that raises the question of how does the virus get in? Because in fact, it can't get through the Paramecium. But I'll tell you in a moment. By the way, these viruses are all over the place. You can find them pretty much in every fresh water around the world. And I love this picture. This is a plaque assay. They have a lawn of the algae alone, just the algae. They can grow, make a lawn, and they can infect it and makes beautiful plaques. Look at that. It's green. You don't even have to stain it. I love it. This is great. Beautiful plaques, too. All right, so how does this virus replicate? It turns out there's a predator involved. So the problem here is that virus particles are excluded from paramecia, right? And how do they get in? Well, they found that the viruses, here's chlorovirus in red, and these are the cilia of the Paramecium, which are shown in A. This chloros stick to the Paramecium, and they're eaten by a predator, which is a copepod, right? One of these intermediate level above the primary producers, swimming around in fresh waters, eating things. And then other things eat them, of course. So the copepods eat the paramecium, and they digest them. And out in the feces of the copepod come the broken paramecia, and these algae are released, and then the virus can infect them. That's how you make viruses. That's how they come out of the paramecium. And presumably some of these infected algae will then be taken up by new paramecium, and they'll grow and so forth. So a predator is involved in getting the virus to be able to infect other cells. So that's virus ecology. We have barely scratched the surface here. I just wanted to give you an idea of what's going on out there outside of our human-centric virology course. I think it's very cool. As you can see, there's not a lot of mechanism. It's mostly descriptive at this point, but it's an early field. Marine biology obviously is very old, but marine virology and freshwater virology is relatively new. But it's going to change every year. I look forward to going to that ChoanoVirus meeting every other year and learning more and bringing it back to my course. I've just given you a sampling of the interactions that we know about. It's, as I said, it's a growing field, and metagenomics has really pushed it forward. But we do need a transition to experimental science. So the growth curve is an example of that. You have to start doing experiments like that. You have to make model systems and manipulate the virus and the host. And we haven't really talked very much about plant viruses and their hosts, but there's a whole story there. It wouldn't fit into this lecture, but in future years we may add some more of that. And basically I want you to come away understanding that there are viruses of other things besides mammals. And we haven't even talked about insect viruses, of course. And these unicellular inhabitants of the oceans, freshwater, land environments, all kinds of interactions. We're just beginning to scratch the surface. And in the end, we want to know how this all works. How do these live together? How do they benefit? How do they harm one another? And that's the basis of that. But, buried in this is also the ecology of how we mess things up and acquire new viruses. I think that's an important part of any virus ecology discussion. So next time we have 3 lectures left. We're going to talk about HIV and AIDS. This is the only virus that has its own lecture because it's so important in the scheme of the world, as you will see.